Good morning, everyone. It's a pleasure to have the opportunity to present an in depth overview of the total organic carbon analyzer. So, this presentation will explore the working principles of the TOC analyzer, the detailed post combustion analysis process, its diverse applications across various industries, and technical considerations that highlight its advantages and limitations. So, let's begin by understanding what TOC analysis is and why it is vital for many industrial processes. So, what is total organic carbon analyzer? TOC analysis refers to the quantification of organic carbon in a liquid sample, most commonly water. Organic carbon is an essential parameter in assessing contamination levels. So, TOC serves as an indicator of water quality and is widely used to monitor the presence of pollutants in various industries. So, significance in industry. Paras- uh, it has a major role in pharmaceuticals. It ensures compliance with water quality standards critical for injectable drugs and solutions. In environmental monitoring, it detects organic pollutants in natural water bodies and wastewater to support regulatory compliance. In food and beverage industries, it maintains product safety by ensuring the purity of processed water. In semiconductor industries, it monitors ultra pure water systems to prevent contamination during high precision manufacturing. So, TOC analysis is crucial for ensuring safety, maintaining product quality, and achieving regulatory compliance in these industries. With this understanding, let us move forward to explore how a TOC analyzer operates. So, the working principle of a TOC analyzer is a systematic process designed for precision. Here is an overview of the steps involved as shown in the flowchart on this slide. So, each step is integral to accurate total organic carbon measurement, starting from sampling and preparation to oxidation and CO2 detection. Let us examine each step in detail. Okay, so the first step is sampling. The analysis begins with collecting a representative sample. The importance of this step is accurate sampling ensures reliable results by preventing contamination or the loss of volatile organics. And this step is automated. So advanced TOC analyzers are equipped with automated sampling systems to eliminate human errors and improve repeatability. Even a small sampling error could lead to incorrect TOC readings, underscoring the need for precise handling at this stage. In step 2, we have dilution. For samples with high CO2 TOC concentrations, the analyzer automatically dilutes the sample with deionized water. The purpose of this step is that it ensures the TOC concentration remains within the optimal range for detection, preventing saturation of the detector. And its technical significance is that automated dilution systems are highly consistent and allow for accurate analysis of diverse samples from drinking water to wastewater. This ensures that both high and low TOC concentrations can be analyzed effectively. In the third step, we have acid addition. So, acid is added to the sample to adjust the pH. So, this process typically involves phosphoric acid or similar acids that are used to convert inorganic carbonates and bicarbonates into CO2 gas that is added. The purpose of this step is that it isolates non purgeable organic carbon NPOCs by ensuring that inorganic carbon does not interfere with the TOC measurements. With acidification, the analyzer might measure inorganic carbon along with organic carbon, leading to inaccuracies. In step 4, you have sparging. The sparging process purges the sample with carrier gas, such as nitrogen or purified air, to remove CO2 generated by inorganic carbon. The technical role of this step is that it ensures only organic carbon remains in the sample for further analysis. The importance of this step is by isolating NPOC, sparging eliminates any potential overlap between organic and inorganic carbon readings. So, this step is crucial in maintaining the accuracy and reliability of TOC measurements. In step 5, we have injection. So, the prepared sample is then injected into a high temperature furnace using an automated system. The precision of this step lies in the automated injection systems that control the sample volume with high accuracy, ensuring consistent and reproducible results. Modern systems use high precision syringes or valves to minimize the variability. In step 6, we have combustion, which is also called oxidation here. Inside the furnace, the sample is subjected to temperatures ranging from 680 degrees centigrade to 1200 degrees centigrade, often with the aid of a platinum catalyst. So the process is that all organic carbon in the sample is oxidized to CO2. The catalyst enhances the efficiency of oxidation, ensuring complete conversion of organic matter to CO2. So this step is crucial for accurate quantification as incomplete oxidation can lead to underestimation of the TOC levels. Now I'll try to discuss the post-combustion analysis and CO2 detection process. It, start, it starts with the transfer of CO2 to the detection system. So after oxidation, the CO2 is carried to the detection system through heated transfer lines. The carrier gas here is a purified gas such as nitrogen, which ensures no contamination during transport. And the technical design is with transfer lines that are maintained at elevated temperatures to prevent condensation of CO2, which could otherwise distort results. So this step ensures the integrity of the CO2 signal as it enters the detector. In step 2 of the analysis, we have non-dispersive infrared detection, which is called NDIR. The most common detection system in TOC analyzers is the NDIR detector. In the working principle, we have the NDIR detector detects CO2 by measuring the absorption of specific infrared wavelengths by the gas. It has different components like a infrared light source, a sample cell, and a detector. I will now tell the use of these different components. The infrared light source, it emits a beam of IR light that passes through a gas cell containing the CO2. Now in the sample cell, the CO2 interacts with the IR light, absorbing energy at its characteristic wavelengths. Then the detector measures the reduction in the IR light intensity, which then correlates directly with the concentration of CO2. So this method is highly sensitive and specific, allowing for accurate quantification even at trace levels. Then the third step of the analysis, we have the TOC calculation. So finally, the analyzer calculates TOC based on the detected CO2. So measurement timing is very important. that peak CO2 concentration over time is measured, and the area under the curve that you see here is used to compute the TOC levels. And in case of MPOC, the analyzer distinguishes TOC from volatile organics, focusing on only NPOCs, only non-purgeable components, for precise results. Then we will try to discuss the applications and technical insights of TOCs. So, as discussed earlier, TOC analysis is vital in industries such as pharmaceuticals, environmental monitoring, food and beverage, and semiconductors. So, the technical precision of TOC analyzers ensures compliance with stringent regulations and enables process optimization in these industries. In the last slide, we have advantages and limitations. So, while TOC analyzers are incredibly powerful tools, they do have some certain limitations. In advantages, we have high sensitivity and accuracy, with detection limits as low as parts per billion (ppb). And automation also reduces manual errors and increases the overall efficiency. In limitations, we have very high initial costs and maintenance requirements, and the sample pretreatment may sometimes require specific applications such as filtration for particulate laden samples. In conclusion, we can say that TOCs or organic carbon analyzers is a very sophisticated tool that combines precision, automation, and sensitivity to meet the demands of modern industries. Its ability to detect and quantify organic contamination with unparalleled accuracy makes it indispensable for quality control, environmental protection, and process optimization. Thank you for your attention. I am very happy to answer any questions that you may have.